Welcome to Brookings Biology, a PowerPoint video about enzymes. This is the last section in Chapter 2, and we're going to look a little bit closer at those enzyme molecules that we talked about when we talked about proteins. Remember, one of the functions of proteins is they can be enzymes. So we're going to take a look a little more closely at enzymes, and to do that, we have to understand a little bit about how chemical reactions work. Chemical reactions need some help to get them started. If you think about a fire, you can pile up the wood, but it's not going to start on fire. The reaction's not going to start going until it gets that first little spark. You need the match to get it started. And chemical reactions need that little jump start, the little kick in the pants to get it going. And so the energy that's required to get a chemical reaction started, to get it going, is called activation energy. When you activate something, it means you turn it on, get it going, and that's what activation energy does. If you look at this little graph, this is an energy graph showing the change in energy during a chemical reaction. And notice reactants, we're going to start with our reactants, and we're going to end over here with our products. And to get from here to here, there's kind of a little hill that those molecules have to get over in order to make the reaction go. That little hill that they have to climb is the energy that's needed to put into the reaction, the activation energy to get it started. Catalysts are molecules that help chemical reactions happen faster. So look at this little diagram. Here's my original reaction, and there's that hill that the molecules had to get up, the energy that needed to be put in to get it kick-started. Once I get to the top of the hill, the reaction can go by itself. It's pretty easy to roll down the hill. It takes energy to climb the hill, and that's that activation energy. If I run the reaction again with an enzyme, a catalyst present, what it does is require less energy to get it jump-started. It lowers the activation energy. It lowers the hill of energy that we need to make this reaction go. So catalysts work by decreasing the activation energy that we need to get a chemical reaction started. They make reactions happen faster by lowering that activation energy. It's kind of like pushing a snowball up a hill. If you've ever been sledding, you know the easy part, the fun part is the downhill slide. But in order to slide downhill, you got to get to the top of the hill to begin with your sled. And that takes a lot of energy to hike up the hill, to drag your sled. So you got to get up to the top of the hill to put energy in, right? And then slide down the hill. That's the easy part. And that's what activation energy do. It gets us to the top of the hill so the molecules can do their reaction and go faster. In living things, what we call catalysts, remember catalysts make reactions happen faster. In living things, the catalysts that do that are protein molecules. That's one of the functions of proteins, is they can be catalysts to speed up chemical reactions. They're called enzymes. Enzymes are catalysts that work in living things to make reactions go. If you look at the little animation, you have an example of a reaction that's using a catalyst. In this case, the big green molecule is the enzyme. And the little pink molecule is going to be our reactant. It's going to join the enzyme. Notice it fits perfectly. The reaction happens. Out come my products. That little pink molecule is splitting into two pieces, two products. And the enzyme is helping that happen. Notice they fit perfectly together like a lock with a key. It's going to help the little substrate do the reaction and kick out the products. Notice the enzyme, though, didn't change. The enzyme is helping the reaction go, but it's not part of the reaction. We're going to stop here. I'm going to ask you to stop the slideshow and go back to where the videos are, where you got this started, and look in the video links. And here we are, enzymes. This is where you started your PowerPoint video, and it says enzyme video. Watch this during the slideshow. I want you to take a break from this video, go back and watch the little video about how enzymes work. and then come back to the PowerPoint. Welcome back after watching your video. If you have not done that yet, please go back and do that. And we're going to keep going in our slideshow. We're going to put some labels on some of the parts that are going on in this reaction. And so first of all, we have an enzyme. The little green molecule is our enzyme. And on that enzyme, there's a place, perfect match, where the substrate is going to sit to do the reaction. It's called the active site. That's where the action is going to happen. Here comes my substrate now. The substrate is the molecule that's going to react, and it's going to fit into that active site just like a lock with a key. Notice it's a perfect shape match. So I'm going to make a combination molecule, part enzyme and part substrate, and the name tells you that. This is what we call an enzyme substrate complex. It's part enzyme and it's part substrate. It's a kind of a combination molecule. Now the reaction's going to happen, the enzyme's going to help it go, and here comes my products out. And notice I still have my enzyme. My enzyme is unchanged, and enzymes are reusable. If you look at the enzyme at the end of the reaction, it doesn't look any different than the enzyme at the beginning. Enzymes can be reused over and over. They can help this little substrate do the reaction and release products, and then it can help the next one come in and do the reaction and release products. Enzymes are unchanged by the reaction, so they can be reused over and over and over again. So enzymes fit their substrate like a lock and a key. If you were to take your car key out to the parking lot and try to start any car in the lot, you know that that wouldn't work. It only works on your car. Enzymes fit like locks and keys with their substrates. And remember, we said enzymes are unchanged and they're reusable, so we can use them over and over again. We're going to watch some little animations to see how that works. And we're going to look at why we need enzymes in the first place, how they help, and then we're going to look at some things that can affect how enzymes work and also what happens when they don't work. So if I was to do a little chemical reaction and I wanted to hook these two little blue sugar molecules together, if they are floating around loosely in the cell, they might bounce around for a really long time before they got lined up in the right direction and close enough together in the right way to hook together and make that reaction happen. If I have an enzyme available, remember that active site fits the substrate perfectly. So if I have an enzyme to help, it lines up the molecules, they're facing the right way, they're close enough together to do that, pull out the water molecule and make a bond. So enzymes help the molecules get in position, and so it speeds up the reaction. It goes faster if you have an enzyme, right? Can make the bond and do the reaction. Enzymes are pretty specific. If you look at these two enzymes, which one of these do you think is going to help this molecule do a reaction to break apart? If you look at the active sites, different enzymes have different active sites, and so they're very specific. They only work on one kind of molecule. Not all enzymes fit all molecules. This one is a perfect fit though, and the reaction can happen. If you look at that other enzyme, that's going to fit a different substrate. It's going to do a different kind of reaction. And so there are enzymes for all the reactions that are happening in your body, and different enzymes fit different shapes of molecules. If you think about the reusable part, my enzyme can do the first reaction, make the bond, and notice it can be reused over and over again. It can take the next molecule and make the bond, and the next, and the next. Enzymes are reusable. When we do a polymer, make a big polymer, we can join those little subunits over and over and over again. Sometimes things interfere with how enzymes work though. Remember, what makes the enzyme able to help is that active site shape matching the substrate. has to match perfectly in order for the reaction to go. If I do something that changes the shape of my enzyme, changes the shape of my active site, remember enzymes have 3D shapes. If I do something to change the shape of the enzyme, for example, heat can change enzymes. Now the substrate won't fit in the active site anymore, and the reaction can't happen. What are some things that can change the shapes of enzymes? One of them is pH. Remember we talked about pH, H for hydrogens. You're measuring hydrogens, and the more hydrogen ions you have, the more acid a solution is. So changing how acid the cell is, changing temperature in the cell, can cause those proteins, those enzymes, to change their shape. They unwind, or we say they denature. They lose their 3D shape. If you have ever left CDs or in your car in the hot sun, you'll know that they will actually warp and change shape because of the heat. And the same thing happens with enzymes. We're going to watch a little movie about that happening.
Can change the structure of proteins.
Let's start that again. I kind of had my sound off.
An egg provides a good example of how heat can change the structure of proteins. Proteins are found in both the egg white and yolk, and these proteins have different structures in raw versus cooked eggs. Let's consider the proteins in an egg white. In their native uncooked state, the proteins are chains of amino acids that fold into complex shapes. Several types of bonds form between amino acids in a chain, stabilizing the globular structure of the protein. When the egg is cooked, the heat makes the molecules vibrate. The bonds linking the amino acids in chains are strong covalent bonds that withstand the heat. However, most of the bonds that stabilize the three-dimensional structure of the protein are weak bonds, typically hydrogen bonds, which break, allowing the proteins to unravel. When two unfolded proteins make contact, they form new bonds with each other. A large net of interconnected proteins forms, and the egg hardens. Light can no longer penetrate the egg, which is why the clear portion of the egg turns white.
So denaturing, unwinding those proteins, changes the shape of the active site, and it makes it so the enzyme can't bind the substrate anymore, and it makes it so they can't help the reaction go. It's one of the reasons homeostasis, that maintaining that stable internal condition, is so important in cells. Because if you make it too acid or too salty or too hot, it causes the enzymes, the proteins that are in your cells, to unwind, to denature, and they lose their ability to function. They can't do their jobs anymore, and that can make you very sick, and that can make you dead. The last thing we need to do is talk about how we name enzymes, because many enzyme names end in the letters ASE. It tells you that word, if you see a word with an ASE, it tells you that molecule is probably an enzyme. Do all enzyme names end in ASE? No, but majority of them do. It's a clue that it's an enzyme. And the beginning of the name tells you what it does. For example, we're going to learn about an enzyme this year called DNA polymerase. The ACE at the end of the word tells you this is an enzyme. And the beginning of the name tells you what it does. It's making a DNA polymer. Remember, joining those little monomer units, those little nucleotide units to make a DNA. The enzyme that does that is DNA polymerase. Right now, if you have food in your stomach and in your digestive tract, you have protease enzymes that are working to break down the proteins. Their ACE at the end tells you it's an enzyme. Another enzyme we're going to learn about this year is ATP synthase. There's that ACE again, and the beginning name of the name tells you what this enzyme does. It's synthesizing, it's making ATP. Remember we talked about our energy storage molecule, that ATP, and adding the P's onto ATP is what charges up that molecule, what stores the energy. And the enzyme that sticks on the P's is ATP synthase. Thanks for watching the slideshow, and stay tuned for our next review slideshow.